Okay, so the following part, after the corpuscle, we will have the proximal convoluted tubule. The proximal convoluted tubule. Now, all of that fluid collected by the Bowman's capsule will be drained into the convoluted tubule. Here, we will have a lot of absorption happening. We have simple we will have simple cuboidal cells with microvilli to increase the surface area and therefore maximize the solute absorption and secretion. So we are going to absorb all of these ions. We will absorb glucose, amino acids, protein, vitamins, lactate, urea, uric acid. All of them are going to be reabsorbed in this area. But also there is some secretion. When we are saying absorption, just let's establish this. When we are saying absorption, it means that we are taking it out of urine. And when we are saying secretion, putting in urine. So let's keep this in mind. Absorption and secretion. Absorption, it means we are taking it out of the urine. Secretion, we are putting it back into the urine. So here, all of these are being reabsorbed or absorbed, and all of these are being secreted there. Sometimes this is active transport, ATP, because we are moving all of these ions most of the time against their concentration gradient. Here it's the area of high concentration, and outside the BCT is an area of low concentration. So we need to use ATP. From there, the filtrate will keep going through the nephron loop. In the nephron loop, it's made out of two things, the descending and the ascending. With the descending, it's solely responsible for absorbing water. That's it. Only. It absorbs water. Whereas the ascending loop is absorbing sodium, potassium, and chlorine. There is some secretion of urea into this loop, but mainly we have two major absorption of water and ions there. Now, the thickness of this loop, it changes because the nature of the cells. Here it's cuboidal cells and here it's squamous cells. That's why we have different width of the loop. After that, we will have the distal convoluted tubule, the DCT. And the DCT, usually it's shorter, but also again, we will see some secretion and absorption. So absorption, we will have sodium, chloride, bicarbonate, water. Secretion, we will have hydrogen ions, potassium ions, ammonia. And the BCT would lead to the collecting duct. So this is the collecting duct. In the collecting duct, mainly we are going to see absorption of water. However, here with the DCT and the collecting duct, we have a different mechanism for absorption, because this absorption is happening here under the influence of hormones. With the BCT and the nephron loop, it's based on the osmolarity of the area around these tubes. But here it's more controlled by the hormones of the body. That's why here it's more important. We'll spend some time on it. Okay, comparing to the BCT and the nephron loop. So for example, on the collecting duct, we have water channels. These water channels will open and the cells of the collecting duct will display more of these water channels once ADH is secreted. Remember the antidiuretic hormone secreted by the pituitary gland? So we said that hormone targets the kidney. So here it targets the cells of the collecting duct and asks them to display aquaporins. These are channel proteins for water. So when ADH is secreted, we are going to reabsorb or absorb a lot of water. And that's how we are blocking urine formation. That's the ADH. This is something we already talked about, and I think I'm going to stop here. The coming video will talk about the physiology of the urine formation.